Hello dear students, how are you all? Welcome to One Page Biology. This is Praful. I hope you all are doing fine. In today's video, I'll be covering a very important concept from the chapter Excretion and Osmoregulation. Dear students, this chapter is from 11th standard CBSE as well as from other state boards. So this chapter is really very important because the questions from this chapter have been asked in the board exams as well as the questions are been asked in the NEET exam as well. So this is a very important chapter. So please try and understand this particular concept. So let's begin with the question. So now the question which is often asked from this particular chapter is nothing but describe the process of formation of urine in humans. And this particular question is always asked for around 4 marks. So let's try and understand the answer to this question, dear students. Now dear students, as we all know that urine is one of the most important excretory products which are formed in our body, and where exactly is the process of formation of urine occurring? So as you can see in the screen, this is the most important excretory organ of our body, which is nothing but kidneys. So we all are blessed with a pair of kidneys. So where does the urine formation occurs? Yes, my dear students, it is inside the kidneys. So the process of formation of urine occurs in 3 major steps, which are nothing but the first step is ultrafiltration, which is also known as glomerular filtration. The second step is called as selective reabsorption, and the third step is called as tubular secretion. So let's try to understand, dear students, one by one, what exactly is ultrafiltration, what is selective reabsorption, and the third, that is what is tubular secretion. So now dear students, the very first step, which is called as glomerular filtration, also known as ultrafiltration. So what exactly is glomerular filtration? So dear students, inside the kidneys there are many tube-like structures which are called as nephrons. Now dear students, the blood actually enters via a particular artery which is known as renal artery, and it further forms something called as afferent arteriole. Now afferent arteriole, here the word afferent means coming in. So the blood enters via the afferent arteriole and it enters inside a network of capillaries which is known as glomerulus. So as the blood enters inside the glomerulus, it is going to get filtered because glomerulus contains many pores, because as we said, it is nothing but a network of capillaries. And then the remaining unfiltered blood is going to come outside with the help of an efferent arteriole. Here the word efferent means coming out. Let me ask you a particular question. Do you think that is there any pressure or force created inside the glomerulus when the blood is actually getting filtered? Yes, dear students, you must have guessed it right. The answer is yes. Yes, there is some pressure created. So what is this pressure? So if you carefully observe, the diameter of afferent arteriole is slightly larger in comparison to the diameter of efferent arteriole. So because there is a difference between the diameter of afferent and efferent arteriole, there is a pressure which is created inside the glomerulus. So what is this pressure called? This pressure is known as glomerular hydrostatic pressure. And how much is this? This is around 55 mm of mercury. At the same time, are there any other pressures which are also created in the glomerulus? Yes, dear students. So there are 2 pressures which are present inside the glomerulus. What are these pressures? One is the osmotic pressure. What is osmotic pressure? It is the pressure which is created because of the solutes present inside the blood. Solutes like, for example, the blood cells, the mineral ions, etc. So all these solutes actually provide resistance to the flow of the blood. So in a way, it is kind of a negative pressure. So this pressure is called as osmotic pressure. And how much is this osmotic pressure? It's around 30 mm of mercury. At the same time, there is some pressure which is created by the blood which is previously present inside the glomerulus, and again this is a negative pressure because it provides resistance to the flow of the blood. And what is this pressure called? This pressure is called as capsular pressure. How much is this? This is around again 15 mm of mercury. So now if you calculate the net effective pressure, so the net effective pressure will be equal to the glomerular hydrostatic pressure minus the osmotic pressure plus the capsular pressure. So if you calculate how much is this, exactly this will be coming around 10 mm of mercury. So the effective filtration pressure is equal to around 10 mm of mercury. So there we go guys. So we are done with the first step of urine formation, which was nothing but ultrafiltration, which is also known as glomerular filtration. Now, once the blood is getting filtered inside the cup-shaped structure, which is called as Bowman's capsule, what is that filtrate called? This filtrate now will be called as glomerular filtrate. Also remember, dear students, such a filtrate is also known as primary urine. So this particular filtrate is actually devoid of the blood cells as well as the proteins. That means the part which is getting filtered is only the plasma, which is the liquid portion of the blood, and the proteins and the blood cells do not get filtered, right? So this particular glomerular filtrate is going to get further passed into the next part of the nephron. What is the next part of the nephron called? The next part of the nephron is called as PCT. What is the full form of PCT? PCT stands for proximal convoluted tubule. So here the word proximal means first, convoluted means coiled. So this is the first coiled part of the nephron. What exactly happens in this particular part? In this particular region, dear students, let me tell you, there is some important reabsorption of solutes which is going to occur in this particular structure. That means the solutes, like for example the amino acids, the solutes like glucose, sodium, chloride, etc., these are all going to get absorbed from the PCT. At the same time, the very important biomolecule, which is nothing but water, nitrates, these are also going to get reabsorbed from the PCT. But remember, dear students, when we talk about absorption here, there are two types of absorption taking place, that is active and passive. So when we talk about the absorption of solutes like glucose, amino acids, salts, these are absorbed in an active manner. That means they require energy, that is ATP, for the absorption. Whereas if you talk about the absorption of water and nitrates, this is passive absorption and this does not require ATP. So if you talk about how is this exactly absorption occurring, so inside the PCT there are certain cells which are present, and these cells are nothing but the columnar cells, and the columnar cells have got many finger-like projections which are present at their surface. These are known as microvilli. They are also commonly called as the brush border. So the microvilli help in the absorption of some important solutes which are present inside from the PCT back into the blood. So this entire process is called as selective reabsorption, dear students. So now we come to the third step. So before the third step, let me also tell you, now once the glomerular filtrate passes into the U-shaped structure, what is this U-shaped structure called? This is known as the loop of Henle. Now dear students, the loop of Henle consists of the two limbs. One is nothing but the descending and the other is the ascending limb. Now if you compare the diameter of this descending and ascending limb, you can very well see that the descending limb is slightly much thicker in comparison to the ascending limb. The reason is the descending limb is more permeable to the water, whereas the ascending limb is permeable to salts. So now from the descending limb there is also further water reabsorption going to occur, as well as from the ascending limb there are certain salts which are going to get reabsorbed. Now dear students, once the filtrate reaches to the next part, that is DCT, that is DCT stands for distal convoluted tubule, and now further after DCT the filtrate will further enter into the collecting duct. So what exactly happens in this DCT and CT? So here comes the third step, that is nothing but tubular secretion, also called as augmentation. So in this particular step in DCT or collecting duct, what happens is some important waste like creatinine or potassium ions or hydrogen ions, they are going to get secreted back from blood inside the lumen of DCT and CT. That means this is opposite of selective reabsorption, right? So the ions like potassium, hydrogen, as well as certain important drugs like some important antibiotics, so they are all going to get secreted back inside the DCT or CT, and then finally the filtrate reaches to the CT, that is the collecting duct. From the collecting duct, again some amount of water as well as certain important salts, they are going to get reabsorbed. That means the urine which finally comes out of the collecting duct is highly concentrated, and this ultimately urine is going to go into the ureters, and from ureters it will go inside the urinary bladder. So dear students, if you recollect, as we said, the process of formation of urine occurs in three steps, which were nothing but the first was ultrafiltration, or also known as glomerular filtration. Second was the selective reabsorption. Third was the tubular secretion, also known as the augmentation. So dear students, I hope you understood this concept, and you have to mention this in your exams whenever this particular question comes, and you have to make sure that you have to write down the all the three steps very clearly. Okay dear students, so I hope you understood the video. In case of any suggestions or doubts, please mention in the comment section. So this is Praful signing off. See you all in the next video. Take care dear students. Thank you so much. Bye bye. Thanks a lot.